This is Audible Bleeding, the Vascular Surgery Podcast. We're here to help you keep your finger on the pulse.
Welcome back to Audible Bleeding. I'm Jacob Schwartzman. I'm here today with Dr. Melina Kibbe. She is the Colin G. Thomas Jr. Distinguished Professor and Chair of the Department of Surgery at the University of North Carolina and Professor in the Department of Biomedical Engineering. She did her general surgery residency at the University of Pittsburgh Medical Center, followed by vascular surgery fellowship at Northwestern. Prior to UNC, Dr. Kibbe was faculty at Northwestern University for 13 years, where she also served as the Vice Chair of Research in the Department of Surgery and as Deputy Director of the Simpson Querrey Institute for BioNanotechnology. Dr. Kibbe's research interests focus on nitric oxide vascular biology and developing novel and innovative nitric oxide-based therapies for patients with vascular disease. She has funding from the NIH, American Heart Association, Department of Veterans Affairs, the AMA, and various intramural sources. She holds 11 patents or provisional patents, has over 200 peer-reviewed publications, and is also editor-in-chief of JAMA Surgery. Her research was recognized by President Obama with the Presidential Early Career Award for Scientists and Engineers in 2009. We are very excited to have her on the show to talk about her many accomplishments. Thank you so much for joining us, Dr. Kibbe.
Oh, thank you. It's a pleasure to be here.
I'm also here today with Dr. Jessica Ruan. She's one of the lab residents at Rutgers, New Jersey Medical School, who's currently working in Dr. Kibbe's lab, and we're excited to have her along as well. And she's also interested in vascular surgery.
Hi, thanks for having me, Jacob.
So, Dr. Kibbe, one of the things we like to ask all of our guests, just so that the listeners can get to know you a little bit, what got you interested in vascular surgery in the first place?
Yeah, so I am more atypical than the majority of medical students. So, as you probably know, most medical students figure out what they want to do when they're a third-year medical student. But my path was kind of typical of kind of who I am. So I knew I wanted to do surgery since high school because I had scoliosis and I had a Harrington rod put in my back. And I thought what my surgeon did was like the coolest thing ever. And I had no family members in surgery. So that was my first experience with surgery. And I was like, I want to be like that surgeon. So all throughout college, I was thinking it was going to be orthopedic surgery until I realized what orthopedic surgery really was. And then I was more like, okay, general surgery. And what happened was in my first year of medical school, when, we're sitting in big lecture halls and hearing different lectures on topics. So there was one lecture, and this was at the University of Chicago. There was one lecture that was given jointly by a pathologist and a vascular surgeon, and it was on atherosclerosis. And the pathologist is a world-renowned, amazing pathologist, and he was telling the story of atherosclerosis and how it basically starts in the artery. And at the time, it was about the one-hit injury, to the wall, and that initiates the cascade. And I was so fascinated by the whole process. And then what clinched the deal was the last half of the lecture was when Bruce Gewirtz, who at the time was one of the senior vascular surgeons there, he came in and showed us a video of a carotid endarterectomy. And I was watching this video of dissecting out the artery, clamping it, and then literally peeling out the plaque and sewing it shut. And I turned to my friend, and this sounds really corny, but it is so true. I turned to my friend at the end of the lecture and I said, I'm going to be a vascular surgeon. And it stuck. It stuck all through medical school, all through residency. I mean, go figure, all from that one lecture. So I will say my path is definitely a little bit atypical because I've had these events that have really defined, my life and my trajectory.
Well, that's pretty great that you knew that right from the beginning. I have to agree that that's probably pretty atypical. Did you have any mentors along the way that were particularly influential or helped move you into the field? And once you were there, anybody who helped move you along within it?
Yeah, so I would say, as a medical student at University of Chicago, Bruce Gewirtz was absolutely amazing and wonderful and very supportive. And, as a medical student, there were some other surgeons, but in particular, Monica Morrow, who's a breast surgeon. She was my mentor who, helped me figure out where I wanted to apply for surgery and match. When I matched at University of Pittsburgh, which was like the best thing ever, In vascular, I definitely had a lot of mentorship from Michel Macaroon. He's the division chief of vascular surgery there. And the whole division, from literally the day I interviewed there until, I matched and graduated, was incredibly supportive of my desire to pursue vascular surgery. And then a really important mentor for me was when I went to Northwestern. So again, I was very strategic in wanting to go to Northwestern for my fellowship and to stay on as faculty because I very much wanted to be mentored by Bill Pierce. And again, one of the best decisions I ever made. Bill Pierce is a very unique individual, and he definitely has an ability to be able to support others in what their passion and their vision for their career is. And I kind of knew that about him. I'd met him at SVS meetings when I was a resident, and there was just something about him that clicked, and I was like, I want this guy to mentor me. And so it worked out perfectly.
That's great to hear. Do you have any advice for any young medical students who may be looking for a mentor right now?
Yeah, I think it's really important to have mentors and role models. And I would say that you want to not be shy. And you basically, if you find somebody who has a career that is similar to the career you envision for yourself, talk to them, approach them. And if you feel that you click with them and that they could be a good mentor for you, then I really do advise medical students to actually have that conversation and say, let's say they're coming to me, Dr. Kibbe, you have an amazing career. It's the kind of career that I see myself having in the future. Could you mentor me along this process? And actually ask. I find that when any faculty member is asked to be a mentor, who's going to say no? It's such an honor. Now, the only other advice is don't ask the busiest person in the room. So I will have to say that I have garnered quite a few administrative responsibilities of late. And so, for example, I may not be the best person to mentor, quite a few medical students where we have a whole bunch of other faculty that would be great. So you do need to be cognizant of who you're approaching and if they have time. But by all means, approach people.
Well, that's really interesting to hear about. I'm also really curious to find out along this unique path that you described, at what point you became interested and involved in basic science and translational science research.
Yeah, so that's very similar to my story about why I'm a vascular surgeon. So when I was a undergrad at University of Chicago, I did the classic thing where during one of the summers, I went and worked in a basic science lab, and it was a neuroscience lab. And this was my first experience doing basic science research. And I got to tell you, it was amazing. I feel like I was bit by the bug, if you want to say. I was given this opportunity to join this lab, and we were basically researching at the time a protein that we knew nothing about, but now we know a lot about it, BNP, brain natriuretic peptide. And so some of this early work is, as an undergrad, I was asked to basically dissect out the circle of Willis from the rat brain using a pretty significant microscope. And the reason I was asked to do this as an undergrad is kind of funny. It's because I had not yet started drinking coffee, and I had very steady hands. And all the other people who were in the lab, the postdocs, this is not a surgery lab, they basically were not able to dissect out the circle of Willis and keep it intact because it is tiny in the rat brain. So it was validating for a couple reasons. So I was able to go in there and do this small animal surgery and dissect out the circle of Willis and keep it intact. So it validated that I did want to be a surgeon. And then at the same time, I did all the immunohistochemistry to really map out the distribution of BNP in the circle of Willis as well as in the heart. We found very unique patterns of where BNP was distributed. So that whole discovery process and then, writing it up and publishing the results. I was bit right there. And it was something that I knew I wanted to do actively going forward because I wanted to be able to have an impact, not just the impact we have on a patient by patient basis, because that is amazing, but I've always wanted to have a bigger impact on improving the way that we deliver care. And you can do that through basic and translational research.
I know as far as some of the research that you've published in Vascular that you have addressed novel delivery methods of nitric oxide delivery that have shown some promise in reducing neo-intimal hyperplasia. Can you discuss the role that nitric oxide has in the regulatory neo-intimal hyperplasia and why the delivery methods you developed are optimal for this delivery?
Great question. So I am a firm believer of the benefits of nitric oxide in the vasculature. Our endothelial cells, our normal, healthy endothelial cells, produce nitric oxide, and they produce it for a reason. It is an amazing molecule. It basically promotes vascular health and keeps all the badness in check, right? And so what do I mean by the badness? So nitric oxide inhibits platelets. It inhibits, all the inflammatory cells. It inhibits the growth of the cells in the wall that contribute to neo-intimal hyperplasia and restenosis. So meaning it inhibits smooth muscle cells, it inhibits adventitial fibroblasts, it inhibits all the bad stuff. And then at the same time, nitric oxide actually supports the growth of endothelial cells, which is what we need, and it's a potent vasodilator. So for all of these reasons, I am a true believer in the benefits of nitric oxide in the vasculature. Now, I'll tell you that my path for research has been a very interesting path. So when I did my 3-year postdoc research at Pittsburgh, I did a lot of gene therapy. And I was delivering the iNOS gene. So if you deliver the gene, it then produces the nitric oxide and has the beneficial effect and prevents restenosis. When I started my faculty career, I very much knew in my heart of hearts that, and this was, in the early 2000s, that I did not think gene therapy was at the point where it was going to be close to being translated into patients anytime soon. So I made the very conscious decision that I wanted to develop pharmacologic methods of nitric oxide delivery. So when I started my lab, I started collaborating with a lot of folks from like chemistry, material science, biomedical engineering, you name it. And it's through these collaborations where I think some of the most, innovative ideas from the lab have been born. So some examples of the things that we're trying to develop are a novel drug-eluting vascular graft. So think about that. What do we have in the field of vascular surgery? We have our prosthetic grafts. And we have the heparin-bonded prosthetic graft, but that's it. We don't have any drug-releasing vascular grafts. So one of our projects is trying to develop a drug-eluting vascular graft that does not simply, elute nitric oxide per se, but it's a polymer-coated ePTFE graft. And we're capitalizing on the mechanism that leads to nitric oxide release. So what we're doing is we're trying to immobilize ascorbic acid onto the graft. And so by doing that, the ascorbic acid is then exposed to all the circulating blood elements and will react with what we call S-nitrosophiles, which we have in our blood. And every time it would react with circulating S-nitrosophiles, it would release nitric oxide at the blood material interface. And it could do this indefinitely as long as the ascorbic acid is immobilized on the graft. So that is one project, and that's a project that's funded by, right now it's funded by the VA, so we just got funding for that. So another project that we have is we basically took a step back and we asked how could we deliver nitric oxide to the site where it was needed to deliver the nitric oxide and prevent neo-intimal hyperplasia and restenosis. So about 8 to 10 years ago now, there was a movement, as we all know, in the cancer field to try to develop targeted therapies for cancer. So in talking with one of my collaborators, Sam Stoop, we basically said, well, come on, we've got the vasculature at our disposal. So we should be able to develop a true targeted therapy that you basically just inject into a vein, and the delivery vehicle will circulate in the bloodstream and then target and bind to the site of interest. And once it binds, it then releases a drug. So we decided we should be able to do this. We should be able to develop a therapy like this and target it to the site of vascular injury and have it release nitric oxide to prevent restenosis. This would be a game changer and a paradigm shift in how we're trying to prevent restenosis. Because to remind you, what do we have now? So we have drug-eluting stents, which deliver drugs at the site of the stent, but that's about it. That's about all we have. So we started on this idea, and I kind of call it my Star Trek project, and it's basically we use a peptide amphiphile nanofiber. So these things spontaneously form a nanofiber-like shape. And what we did is we targeted this nanofiber to collagen. Because after you do something like balloon angioplasty or even just sewing, a vein onto the artery, what do? When you're injuring the artery, you're exposing the underlying media, and the media has collagen. So we said, all right, let's use collagen as our target. So we added a little peptide sequence onto this nanofiber that will target it to collagen. And sure enough, when we would inject this nanofiber into the tail vein of rats after we did the balloon angioplasty to the carotid artery, this nanofiber actually circulated in the bloodstream and then bound to the site of injury, and only to the site of injury and nowhere else in the vasculature. So we were like, oh my gosh, this could really work. So then we attached nitric oxide to the nanofiber, and then same thing, showed that it bound to the site of injury, and at 2 weeks, it completely prevented myointimal hyperplasia, and it was durable out to 7 months. So that is a project we are still actively working on, and we're now working to translate that into an atherosclerotic environment. But that project has borne many different targeted projects that include trying to target hemorrhage, trying to target lung tissue and inhalation lung injury, trying to target pulmonary hypertension, and again, kind of the lung tissue in that, and aneurysms. We've most recently also started on trying to target aneurysms. So that's been a suite of therapies, and I'm really excited about where that's going.
Yeah, that's all very fascinating. I was wondering if you can talk a little bit about some of the challenges that come with conducting translational types of research and the steps that are necessary to bring such a promising therapy to human testing.
So I think the challenges and the steps are kind of almost one and the same, in my opinion. I think it's really important to collaborate. So even in what I just described, you could see that with all of my projects, it requires a lot of expertise from a lot of different disciplines. And I personally credit any success that I've seen with my research, I've credited to the fact that I have amazing collaborators that I learn from constantly. And it pushes me to be better, and it pushes the science to be better. And some of the most innovative ideas that I've developed it with collaborators. So I would say that is the challenge, is overcoming the concept that you can do it alone, because I really don't think you can do it alone. You need help and you need expertise from a lot of people. Now, the steps involved in translating the research to humans is a whole nother topic. It's not easy because it involves, the whole pipeline of drug discovery and development, if you want to think of it. So the pipeline is this, that you're typically starting to develop your therapy and you're looking at it in cells, and then you're going to translate it into animals. And if your animal studies are successful, then you're going to translate it to clinical trials. Then you need to go through your phase 1, your phase 2, and your phase 3 clinical trials before you're actually able to get something FDA approved for use in humans. So it's a long road and a long path, and that too requires a lot of expertise from a lot of different people to navigate the waters of getting through the clinical trials and also getting through the FDA process to getting it to humans.
That's great. I understand it sounds like it can get frustrating at times, but overall can be really rewarding.
It can be very rewarding, and you just have to have perseverance.
Which it sounds like you definitely have.
Yeah, I don't know why, but I do.
Dr. Kibbe, thank you very much for that. It was very insightful, and it's great to hear about some of these new therapies you're developing. I was lucky enough to be at the grand rounds that you gave last year at Rutgers, New Jersey Medical School, where you talked about biocompatible elastomeric polymer-based customizable stents that you're developing. Can you tell us a little bit about that project and what you hope to accomplish with that, and maybe some of the history behind what we've tried to do in this field previously?
Yeah, so this is a project that also was born from a discussion with one of my collaborators at the time, Guillermo Mir. So he's a biomedical engineer. And it's really interesting how we started this. So literally one day, because we shared an office, we were sitting in our office, our research office, and Guillermo was like, let's talk about some of the challenging areas in vascular surgery. I said, all right, well, listen, we still have a challenge with when we balloon angioplasty arteries, and the bare metal stents that are put in still lead to significant rates of restenosis. So this still is a problem. And that then leads to, recurrent procedures on patients and inevitably possibly bypass, et cetera. So Guillermo asked me a basic question that, I was so close to, I probably never would have asked myself. He said, well, Melina, why do you guys put metal in arteries? So I said, very good question. The only reason we're putting metal in arteries is that's what was developed first. And so we put the metal in, and we really put the metal in for two reasons. One, to prevent that elastic recoil that occurs right after you do a balloon angioplasty, because, right, the artery wants to go into vasospasm. And then two, we put in the metal stents because you want to basically tack down the intimal layer so that you don't get a dissection. So those are the main reasons why metal stents were first introduced. So I told Guillermo that, well, we don't actually need the metal. It could be other materials. And at the time when he and I had this discussion were some of the early biodegradable arterial stents. And these were basically made out of polymers. But these are preformed, matrix design biodegradable stents. And so there were many challenges with that kind of concept of a preformed matrix, design polymer stent that kind of looks the same as the metal stent. And the challenges are this. Imagine you have this polymeric stent. You have to collapse it into a tiny little delivery device. It needs to be flexible. You need to be able to snake it around the body to get it to the site that you want to deliver it. And then deliver it, have it expand, have it stay intact, and have it have enough external, resistance to prevent the recoil. And then, of course, deliver the drug. So there were companies working on this early on. And in particular, one of the early ones was Abbott. But indeed, they were facing a lot of challenges. So Guillermo and I said, well, can we rethink how to actually stent an artery that could overcome all of these challenges? So that's where we came up with our idea, and we call it our liquid stent project. But basically the idea is that after you inflate a double occlusion balloon catheter, so you're isolating the segment of artery that you want to cast the stent, the idea is we would inject into this space a liquid polymer that we would then inflate an inner balloon that would cast that liquid polymer, forcing it up against the wall of the artery. We would then send down a laser light that we would turn on for a couple of seconds. And you turn it on, and it would turn that liquid polymer into a solid polymer, casting the stent. And then you would take the laser light out, you would deflate the balloon, rinse out the space to get any unreacted polymer, deflate your occlusion balloons, and you're done. And now you've casted, I would say, a designer stent, or you could go as far as saying it's a personalized stent that was shaped to the contour of the individual artery, completely coats the surface of the artery. So that's important because it has the greatest ability to prevent platelets from adhering, and it has a very large surface area for delivering a drug. So that's the idea of our liquid cast stent, and we've been working on this for about, anywhere from 8 to 10 years now, in really kind of fine-tuning this. And we're at the point where we're now testing it in pigs, and we are delivering nitric oxide as our drug. So it's really exciting. And again, this would be a paradigm-shifting way to stent an artery.
That's so incredible. You mentioned a double occlusive balloon with obviously some suction ports and a polymer-based delivery system. Can you talk a little bit about how you developed that? Because obviously that's not on the market right now.
Yeah, and it's been very challenging, actually. I have experienced a lot with developing this catheter, and to this day, I'm still experiencing a lot. I've learned a lot about how to interact with industry partners. So, I'm a vascular surgeon. I'm an active practicing vascular surgeon. So I had a very good idea of the specialty catheter that this project would need. So when I first was working with a company, I explained to them the concept, and I had kind of drawn out, rudimentary drawings of what I thought we needed. And it was fascinating because the first company that I worked with, even though I had very specific ideas of, the French size, the materials, how many lumens I needed, what I needed to do, we still had to go through this whole big matrix of all these different options to evaluate everything, only in the end to come back to what I originally pitched. So that was kind of funny. So then I went through a series of working with companies, and I had not the best experience, I will be honest, because this is a very challenging catheter to fabricate. So much so that some of the granting agencies that I did receive funds from in their early grant applications, when I was denied the grant, it was because I didn't think I could get the catheter made. And so once I finally did get the money, again, it's all about perseverance and learning how to work with the company. I am now on what I would call prototype version number 4 with my 4th company. And it's actually at the point where the catheter now is pretty good and it's functioning fairly nicely. We're still needing to tweak different aspects of it. And so I've got an order right now that's going to be fabricated shortly.
That's pretty incredible. And now that you have the catheter made, you mentioned testing this in pigs. What are some of the next steps and some of the challenges you anticipate moving forward with this idea?
So we're at the point now where we're more consistently, we just did a series of multiple pigs just over the last couple of months, and we're still trying to fine tune basically how much laser light is needed when we do this in a live animal, the energy required to polymerize this, and for how many seconds. And we need to do this also in a safe manner with the chemistry of the nitric oxide release. It's a little challenging because the nitric oxide counters some of the beneficial effects of our laser light and the catalyst we're using. So it's a little bit more complicated than you probably want me to get into right now. But these are kind of the things that we're having to work out and troubleshoot on the bench. And then we'll be back in the animals again.
That's pretty incredible. Even to come up with a concept like this, I'm sure it took years of hard work. And if something like this did come to market, it would totally revolutionize the entire field of endovascular surgery. So we congratulate you on your hard work and thank you for sharing that with us.
To switch gears, I wanted to touch on some of the research you have done looking at sex bias in surgical research that you demonstrated is prevalent from all the way from bench research to clinical testing. How was it that you originally discovered this was an issue in the research community?
Yeah, so this is a really important topic. It's not, my own research per se, but it's a topic that is very important to me, and it should be important to all investigators, physicians, and even patients. So I first kind of became aware of this when I was at Northwestern, when somebody who's now very much a role model and mentor for me, Teresa Woodruff, she brought it to my attention. So she's the director of the Women's Health Research Institute at Northwestern. And she was leading a group of us where she basically said, listen, this is a problem. And the problem is this, that most investigators, when they're conducting their research in their labs, are developing and testing their research in male cells and male animals. And so what happens is if you're developing and testing these therapies in mostly male animals and cells, you don't know how it's going to react in the female animal and cells. And when you take the therapy to the clinical trial stage, it has not been adequately vetted in both sexes. And so one of the problems here is that you can have the development of drugs that actually get to the market that could have very significant side effects in women because, again, it has not been fully developed and tested in females all the way at the beginning stages of the pipeline, from cells to animals and then into humans. So it's a big problem. I will say most people are not aware of this. 80% of drugs are removed from the market because they have adverse reaction in women. And women suffer very significant consequences of these adverse drug reactions. So if we're ever going to reach a state of true personalized medicine, we have to get this right. We have to get the variable of the two sexes down first. So that's what concerns me.
Yeah, that's really important. What did you find to be the most surprising revelation once you started digging into this topic?
Well, there were a lot of things. So what we did is to look at this in the surgical literature. We, analyzed 5 of our kind of broad surgery journals. And I was surprised to find a very significant percentage of peer-reviewed manuscripts that were published that weren't even stating the sex of the animal or the human that they were researching on. So that was a surprising finding. But then very consistently in the basic and translational research, that people are mostly studying to the degree of 80% of the studies are on male-only studies. And when we looked over time, we're actually getting worse with this practice. So our practice of studying only males when developing our research has gotten worse over time. So those were the most surprising findings.
Wow, that's all very shocking. Other than including and matching both sexes in studies, is there anything else that researchers can do to address these issues?
Well, I think we need to increase awareness, because I still think that there's a lot of people who are not aware that this is a problem and are still studying mostly single-sex studies. So I think we need to increase awareness. I think that investigators do need to make sure that they're developing and testing their therapies in both sexes. It needs to be powered to do so. You know, the pushback is that investigators will say it takes more time, more money, and more animal lives. And that is all true. It does. But it is the right thing to do, and we need to do it because we need to be developing therapies for both sexes, men and women.
Yeah, completely. And was there any response from the NIH or the FDA to your publications?
Yeah, so what happened is it wasn't just our publications that we put out on this. With her leadership, what we decided to do is we knew that in order to move the needle on this topic, we needed to get a national platform for the topic. So what we did is we wrote away to a whole bunch of the different TV news program shows. And what happened was one day the producer for Leslie Stahl actually responded, and she thought this was an important story. And so Leslie Stahl then did a story which aired on 60 Minutes. And it was that story is what started to actually move the needle and have an impact. And it was fascinating to watch. And so we kind of timed our publication to come out around the same time as the 60 Minute story. And what happened is within 3 months of 60 Minutes airing the story about how we're basically developing and testing therapies in mostly males, the NIH came out with their announcement that they were going to develop a policy that was going to require investigators to include sex as a variable in all grants going forward. Now, it took the NIH a year and a half to do that, but they finally did come out with the policy on implementing rigor and transparency. So today, every grant that is submitted to the NIH is scrutinized by the reviewers and whether or not the investigator is considering sex as a variable. And it is a score-deriving factor. So that was good. But if you ask me what else has been done, not much. And so from that perspective, it's disappointing because I would like to see a lot more done, especially by the FDA, because it really is the FDA's job to ensure that they are approving drugs that are going onto the market that are equally effective, or at least effective and not have dramatic side effects in one sex over the other.
Yeah, absolutely. And what requirements, if any, apply to industry-funded studies?
Well, that's also a problem. So right now, the only law that we do have a law that does require that women and minorities be included in clinical research funded by the NIH. But that's what the law is. Clinical research only, funded by the NIH. So any clinical research that is funded by industry is not having to follow that law. So what industry-funded clinical research has to abide to is basically the FDA when they're submitting a new drug application. So again, this is why I think that the FDA really needs to examine their practices to make sure that they're supporting that we have development and testing in both sexes.
Yeah, absolutely. I'm hoping that those changes will happen. Do you have in mind anything as far as next steps from your standpoint for helping move that process along?
Well, I just continue to slowly examine these different issues. So one thing that I'm working on right now is we actually, now that it's been 5 years since I did the original study, so we just took a look and re-examined the same 5 journals to see if we've gotten any better at this process. And so we're analyzing the data right now, and we're hopefully soon going to be writing up that manuscript for peer review publication. So watch for that to come.
Absolutely. We definitely will. What an interesting topic.
Dr. Kibbe, thank you so much. This was excellent to hear both about some of your basic science research and some of the sex bias in research that you've been working on. And you've clearly succeeded as a basic scientist in your career. However, naturally, for a lot of surgeons nowadays, either that's not a possibility or it's an option that's being explored less and less. In fact, in your 2017 Annals of Surgery editorial, you cited declines in basic science abstracts submitted both to the AAS and the SUS from 48% in 2011 to 27% in 2015. And then additionally, as part of a survey you sent out, only 32% of faculty believed that it was feasible for surgeons to be successful in basic science. And so the title of that editorial was Extinction of the Surgeon Scientist, which is a pretty strong statement, but clearly there are people like you who still manage to succeed with it. So what are some of the ways that some trainees and young surgeons who are interested in basic science can sort of keep that passion alive and continue to be successful both clinically and in basic science at the same time?
Yeah, this is a very important topic, so I'm really glad that you asked me about it. I am worried because the number of submissions to the Academic Surgical Congress, which is the meeting you were referring to, that are basic and translational continue to decline even more every year. And so it's reflective of the fact that we have fewer and fewer people that are pursuing basic and translational research in surgery. And listen, if we don't do it, if we as surgeons aren't at the table trying to improve the care that we're delivering through basic investigation and discovery science, who's going to do it? That's what worries me. And it worries me that our discipline of surgery is really going to be negatively impacted by this. So what can individuals do? Well, one, I worry about exposure, meaning that, it's getting harder and harder for departments of surgery to allow residents to be exposed to research experiences. Fewer and fewer programs are allowing residents to take 2 years off to do research. It's costly. I get it. It is not easy to figure out how to fund. But it's important and we need to do it. So what can students and what can residents do? If students and residents have an interest in pursuing basic and translational research, they need to find good mentorship in this area. Because good mentorship, it's going to make the difference and it's going to be able to expose them to the true benefits of doing basic and translational research. You mentioned that survey that only 32% of faculty think it's even feasible. Well, that number is going to also continue to get worse because we're going to have fewer and fewer surgeons out there who've done it. So, of course, if you haven't done it, you're going to think it's extremely hard. It's doable. You just have to have a passion for it, and you just have to do it. And we, as a specialty, need to support it.
I assume that one of the barriers to more surgeons joining the basic science realm, other than finding funding and things like that, are just the time that it takes and the difficulty with balancing that with clinical responsibilities and documentation and things like that. How do we as a medical society overcome this problem and make basic science more attractive to surgeons in the future?
Yeah, well, I want to actually kind of turn that around to you, right? Because there are so many residents now who just don't have an interest in wanting to pursue basic and translational. So I would ask you, why? Why is that? I mean, some of the responses that I get when I ask, medical students and residents is they're told by others it's too hard. They're told by others that to have a career as both a surgeon and a scientist is like ridiculously impossible. It's the hard path to follow. Success is to only a very few. I mean, what are some of the other reasons you've heard?
I think that some of the reasons are people aren't necessarily comfortable conducting basic science research. The lab skills that need to be acquired might seem, somewhat intimidating. And additionally, I feel that a lot of folks are more interested in the clinical research because they are, working with patients every day. And that seems to them much less barriers to overcome to actually get into doing that type of research.
Right, and more of a natural fit. Well, this is exactly what worries me. And so that's why I think we need to make sure that we continue to provide these opportunities for residents and expose residents to these experiences. Because if we don't continue to expose residents, then we're not going to have enough surgeons going into basic and translational research. And that's where we get the extinction of the surgeon scientist.
Yeah, I agree. I think that one of the things that we actually as residents can do, especially those of us who've gone out into the lab and done basic or translational science, is really advocate for it and share and express that it's not that hard, especially compared to all of the other barriers and challenges we've overcome to even get into a surgical residency.
So I could not agree with you more. Let's take you as a great example. I mean, you were able to come into the lab. I mean, surgery residents are very, very smart, and you're able to pick up on things. And if you're doing translational projects that do involve, a lot of animal surgery, you're a great example. You picked up on the nanotechnology very quickly, and you then established your model of hemorrhage with what I consider to be ease, and you were able to validate it and get very reproducible results. It only took you a few months, and you're off to the races. So you're a great example of something coming into lab and picking up an entirely new skill set, but doing it pretty quickly.
Yeah, I mean, it was a great experience, and absolutely having the surgical skills coming in, it actually gives you an edge in a way over folks who come the traditional PhD route. Yes, they have a lot more basic science knowledge and experience, but you work together with those folks and, ultimately you can get better outcomes.
Exactly.
That was great. Thank you so much, Dr. Kibbe. We're just going to wrap things up here with a few questions that we like to ask all of our guests. What is one of the tricks you've picked up over your career that maybe gets you out of trouble in the operating room when you're in a tight spot?
Oh, always follow your gut. Oh my gosh. Yeah, that's easy. So follow your gut. I mean, by and large, we are all very well-trained individuals. And so I have learned to listen to my instinct and take it seriously.
Great advice. One other question. What do you like to do in your free time?
I'm a total nerd. I'm a total sci-fi fantasy nerd. For me, escapism is watching movies. And when I do go on vacation, I will read through some total nerdy sci-fi or fantasy book. So, I basically love anything with the word star in it. So Star Wars, Star Trek, Battlestar Galactica, you name it. If it's got the word star in it, just assume I'm going to be watching it and I'm a fan.
That's pretty awesome. One last question. In the field of vascular surgery, what is something you know to be true, but that others may disagree with you about?
That pursuing a career in surgery is amazing, and you will never, ever regret it.
Well, I don't know that any surgeons would disagree with that one, but...
Oh, but they do. Oh, no, but they do. How often have you met a medical student that was told not to pursue surgery because they would not have a good work-life balance or it's not conducive to having a family? Have you not met medical students who've told you they've been told that?
Oh, of course. I've been told that myself.
Exactly. So that is what I am telling you, something that is true, no matter if other people disagree with me. I think surgery is an amazing career choice, and you can have the work-life balance that each individual person wants. And you definitely can have a family and a significant other and have an incredibly fulfilled life. I love what I do for a living. I could not picture doing anything else, and I am so fulfilled in what I do. And because I'm a surgeon scientist, I could never give up the clinical side, and I could never give up, the science, because I love both, and I love both for different reasons. And it also provides me on a day-to-day basis where I am constantly being challenged and being pushed to be my best. And I get to work with people like Jessica, who keeps me on my game. So it is incredibly fun and a rewarding career, and I can't think of any other career that I would have wanted to pursue.
That's so inspiring.
Dr. Kibbe, thank you so much. That was some excellent advice. Thank you very much for joining us. We really appreciate your time talking about your research and providing some insight on all these other issues.
Oh, it was a pleasure. Thank you, Jacob and Jessica. It was an absolute pleasure to talk to both of you.
Thank you both.